Okay. Very warm welcome to all. At this occasion, we to celebrate the National Science Day organized by Institution Innovation Council, Galgotias University. So, dear participants, you might be aware that you might be aware that 28th February is celebrated as National Science Day in India. But do you know why and for what this day is celebrated as National Science Day? I am also sure you must have heard about that the National Science Day is celebrated as Indian scientist Sir Chandrasekaran Venkataraman. On this day in 1928, he discovered a phenomena of scattering of photons, which was later known as Raman effect after his name. So after 2 years, in 1930, he got Nobel Prize for this remarkable discovery. And this was the first Nobel Prize for the India in the field of science. To mark the discovery of his famous discovery, the National Science Day is celebrated in India on the day each year. In 1986, the National Council for Science and Technology Communication, NCSTC, asked the Government of India to designate 28th February as National Science Day, which the then Government of India accepted the proposal and declared the day as a National Science Day in 1986. So the first National Science Day was celebrated on February 28, 1987. So we also have two eminent speakers, renowned scientists of India and renowned in the globe. I would like to introduce them first. We have Dr. Pallab Bhattacharya sir, and we have Dr. Santosh sir. Dr. Pallab Bhattacharya sir is presently an associate professor, dean and director in charge at NIPER Ahmedabad, Gujarat, which is an institute of national importance. He obtained his PhD degree from IIT BHU and further had his postdoctoral training at University of Miami Miller School of Medicine, USA, under Professor Dilip Yavagal and Nobel laureate Professor Andrew Sely. He was the visiting scientist at Aarhus University and Harvard Medical School, Boston. He has received advanced training in stem cell biology at Marine Biological Laboratory, as well as in the MIT USA, and also in the field of transgenic animal at the Danish Center of Transgenic Mice, Aarhus, Denmark. His present research is to test the safety and efficacy of intraarterial delivery of mesenchymal stem cells in a small and large animal model of ischemic stroke and dissect related mechanism of stem cell mediated neuroprotection with its related imaging specs. Presently, he is collaborating with Nobel laureate Professor Andrew Sely to explore the role of GHRH in ischemic stroke. His research interest also includes regulatory RNA-mediated mesenchymal stem cell engineering and nanoparticle-based targeted drug delivery to the brain and ischemic optic neuropathy. In past, he has awarded with early career grant of DST, Return Home Grant by ISN, Young Investigator Award by Society for Nanomedicine, USA, and Young Neurological Award by the European Neurological Society. So I would like to request Professor Renna Patnaik ma'am to welcome our chief guest. Ma'am, you are muted.
Respected Chancellor, sir, our very dynamic CEO, sir, Honourable VC ma'am, deans, faculty members, students, and of course, Dr. Pallab Bhattacharya and my dear friends. It's giving me immense pleasure to speak few words on this day of National Science Day. And I'm proud to see Dr. Pallav as our guest. He has a very strong and very long bonding with me as my PhD student. Proud to see you here as our guest, Dr. Pallav, and really, I'm proud that you are achieving such heights. And presently, I welcome you.
Thank you.
And I'll speak few words about this. National Science Day, as Dr. Gaurav has already explained, is celebrated every year on the day of February 28th. This day recalls the notable inventions of Raman effect by India's greatest scientist C.V. Raman, and he was awarded the Nobel Prize for this discovery. Since that day, 28th February is celebrated as the National Science Day to recall the discovery of Dr. C.V. Raman. National Science Day is celebrated with a view to spread the message about the significance of science and how it improves the daily lives of the common people. The celebration has following objectives in mind: that display of all activities, efforts, and achievement in field of science, and to provide opportunities to those citizens of India interested in science, and to foster and encourage interest in common folks. The theme for this year is Integrated Approach in Science and Technology for a Sustainable Future. It's launched by Union Minister Dr. Jitendra Singh. It is all about integrated approach for various science and technology organizations and departments so as to support suitable future, that is all the science departments to work in collaboration. The purpose of celebrating the day with this theme is to give community of the country a chance to emphasize their importance in front of our public. Now, the integration of science is based on all science disciplines, engineering, medical, technology, coming together and working in integrated manner. Should involve and initiate industry startups. So, as teachers and students, it is our responsibility to honor the great man and his inventions by contributing greatly to the field of science through innovations, inventions, leading startups for the various industries. So, with these few words, I once again congratulate everybody for celebrating National Science Day. And it is the best time to give push to integrative approach to all the ministries and departments coming together and work for the survival of the humanity at the time of this pandemic crisis. So once again, I welcome Dr. Pallav, and nice to see you here on this stage.
Yes, thank you so much, sir, for accepting our invitation and grace the occasion. So over to you, sir.
So thank you very much, Gaurav. And first of all, my heartfelt regards for the Chancellor, the Vice-Chancellor, the Dean, my PhD mentor, Professor Anjana Patnaik, for giving me the opportunity to interact with you all, to talk about my science before you on this beautiful occasion of National Science Day. So it's been my pleasure that I can tell you that being admired and being appreciated by your PhD mentor, something keep you at the top of the sky. So thank you, ma'am, for your appreciation. And as you have very well mentioned, that the integrated approach in science and technology is very much required. So that what I can say, my lab is working at the National Institute of Pharmaceutical Education and Research, which is an institute of national importance under Government of India. So Gaurav, can I share my screen?
Yes, sir. Yes, sir. You can, sir.
Is it visible?
Yes, sir. It is visible, sir.
So to introduce with, so my lab is working on the stem cell therapy for stroke, and we are trying to deciphering the different mechanisms that usually gives a hint how we can take our therapy from bench to bedside. So first of all, these are my disclosures. My funding partners from Department of Science and Technology, Government of India, ICMR, a startup from NIPER, industrial projects, Indo-US grant, IBRO. So first of all, for all my young friends over here, what actually stroke is? So if you talk about a stroke basically can be categorized into three types. It can be hemorrhagic, it can be ischemic, and it can be a transient ischemic attack. If you talk about the hemorrhagic stroke, it is due to the leakage of blood in and around the brain from a ruptured vessel, which creates a pool of blood around the brain, and it increases the pressure of the brain. It can be due to a brain trauma, brain tumor, arteriovenous malformation, aneurysm. Well, if you talk about ischemic stroke, it usually occurs as a result of an obstruction within a blood vessel supplying blood to the brain. And the reason behind it might be attributed to thrombosis, systemic hypoperfusion, cerebral venous sinus thrombosis, or embolism. Now, if you talk about the American Heart Association, they have come up with some new types of stroke, which we call as a brainstem stroke or a cryptogenic stroke. What is cryptogenic stroke? The stroke which has an unknown cause. You do not know the cause, but the manifestation of the pathology comes to the patient and you don't know the reason. So that's what comes under a cryptogenic stroke. Now, if you look into the pie chart, you see that more than 85% of stroke are ischemic in nature, while 13% are hemorrhagic stroke. As I have mentioned, the different types of stroke, rest all comes in this 2%. So it's the demand of the time that you need to intervene, you need to work on the type of stroke, which is the major killer or the major reason for mortality and morbidity around the globe. If you look into the stroke facts, the incidence rate of stroke in India is in between 145 to 154 per 1 lakh individual a year. On an average, in India, a stroke occurs every 40 seconds, and every 4 minutes, someone dies of stroke. If you talk about the half of all strokes, it can be due to the hypertension that we are having, mostly the Indian populations. 1/4 of all strokes in India occur in people who already had a small stroke before. And this makes the stroke as the third leading cause of death in India. And if you talk about the rural areas, it is the #1 cause of death in the rural India. Now, if you talk about the risk factors of a stroke in elders, you see that those people who are above 85 have more 80% of incidence in stroke in comparison to those between the age group of 74 or 84 who have only 60%. But if you talk about the gender difference, there is a gender disparity in the stroke outcome, where in the age group of 65 to 84, the female are less vulnerable, while the male are more vulnerable. But if you talk about the age group of 85 and above, the females become more vulnerable. Now, what is important? If you have a stroke, you need to recognize whether it's a stroke or not. And you need to rush the patient to a hospital because time is everything for a stroke. So if you look into the facts and if you know the signs of the stroke, it will help the patient to come out of it or may have left functional deficit if he is intervened on time. So if you have any person in your house or someone who faces stroke, you just check whether he loses a balance or not, whether he has sudden loss of vision, whether his face movements are unusual, whether he has a hemiparesis, lightness of one arm or leg, he has a slurred voice, he has speech difficulty. Now it's a time to push that person or take that person to a stroke intervention center where he can be intervened in a proper way. Now, if you talk about the approved treatment strategies, the most important that comes in the Indian scenario is the tissue plasminogen activator. Now, if you talk about the TPA, the limitation is the time window. The patient, if taken to the hospital, if is not intervened with TPA within 3 to 4.5 hours, then it is of no use. The maximum chunk of neurons is dead. The second, the most approved therapy is the mechanical thrombectomy. The mechanical thrombectomy is a mean by you insert a catheter from the femoral artery and take it to the clot area and pull down the clot. But the limitation in the Indian scenario is that this requires a high-end hospital, high-end skill, and even the economical condition of the patient should be good so that he can afford to get a mechanical thrombectomy. Now, you are left with only one administrative molecule, or you can say one administrative protein, that is tissue plasminogen activator, and that to have a limitation of a limited time window. Now, what to do? Being a scientist, you need to think how you can intervene stroke when you have a limited options to treat stroke. So the questions that arise before us, that is it possible to replace the neurons with the neurons? If yes, how? Will it be possible to stimulate the endogenous repair mechanism? If you talk about the endogenous repair mechanism, it may be subventricular zone neurogenesis. Whether you are trying to render neuroprotection, you have to see whether you are able to prevent the ongoing injury or not. And apart from that, the toxic environment that has been triggered due to a ischemic insult has to be modified. Because even if you are going for any type of treatment, this requires a modification of the toxic microenvironment going on the brain, which has to be kept in a halt. So where there comes the concept of stem cell therapy. So what are the stem cells? The stem cells are the cells which has the capacity or the ability to repair, replace, or regenerate. If you talk about the Indian scenario, it has been used for reducing the inflammation. It has been used for muscle regeneration. It has been used in the case of cartilage formation. There are multiple, you can use these cells in a multiple way by just having some factors given to that so that it can transform itself to the type of cells that you want. But the question is that if you're talking about the brain, how much it is going to be helpful? So there are a number of types of stem cells. It can be neural stem cells, embryonic stem cells, mesenchymal stem cells, hematopoietic stem cells, induced pluripotent stem cells. So what do we have chosen? We have chosen the mesenchymal stem cells. What is the rationale behind it? So if you talk about the mesenchymal stem cells, these have a high potential for stroke treatment. Second, if you are going to isolate it, if you are going to take it out, there are no ethical concerns as they are from the non-embryonic sources. Majorly, the most important thing is that these are immunoprivileged, means these have very less MHC1, that is what we call our major histocompatibility complex 1, and this lacks MHC2. And this can be easily produced. Now, when you have a stem cells, so what can be the route of delivery of the stem cells to the patient? So there are various delivery modes that people have tried. These are intravenous, intraparenchymal, intracerebroventricular, and intraarterial. Now, as I have mentioned before my talk, that my strategy is to take the research from bench to bedside. Now, why I have taken intraarterial, this has a little background. So nowadays, when you talk about the FDA-approved mechanical thrombectomy, they usually use the arterial way to push the catheter to the part of the brain and pull down the clots. Now, if you are able to take out the clots by that route, in that case, that route can be easily accessible for pushing those cells to the site of injury. That's what we thought when we started working on this. And apart from that, when you're talking about the clinical implications, it should be less invasive, and this should not cause much discomfort to the patients. So if you talk about the history of stem cells, it didn't come a single day. If you talk about the history of stem cells, so the history came long back in 1957 when Donald Thomas, who was awarded Nobel Prize in 1990, he used the bone marrow cells and he transplanted those between the identical twins of a patient suffering from acute leukemia. And this was the first case of stem cell therapy, although it was not successful. University of Minnesota in 1968 performed the first successful bone marrow transplant, and they reconstituted the bone marrow cells from one sibling and cured the other who was having a skid. So this was, you can say, was the first breakthrough where there was a hint that, okay, you can have a stem cell therapy for intervening a pathology. The major one comes the PC's trial, which was carried out at University of Glasgow, Queen Elizabeth University Hospital. And this was the most and the first successful, you can say, the stem cell therapy achievement that the scientists had. What they did, they gave a single intracerebral dose of neural stem cells of 20 millions. And this didn't produce any adverse effect, and the patient was benefited of the neurological outcome. So this was something which came into the mind that stem cell therapy could be carried out further for intervening different neurological disorders that also may include stroke. Now, starting with my study, or starting with the group where we are working on, we have to have rationalized why we are taking the intraarterial route and why not intravenous. The work came from Michael Chop from University of Michigan, where he pushed 100 million cells to the brain, and he found that 4% of cells entered the brain and survived. Whereas he made a comparative study where he saw that 21% of the cells entered the brain and survived. So this was a distinctive study in 2008 that Michael Chop did. And even I had a chance to talk with Michael Chop when I was in the US, and he was even that time not knowing that whether this study is something which will be a crucial study for taking this from bench to bedside. Now, he had a very good rationale that if you are injecting cells in the intravenous route, what do you call about the first pass? Whether it is going to circumvent the first pass metabolism, yes or no? The answer is no, it cannot. But if you talk about the intraarterial delivery, it can easily circumvent the systemic circulation. Now, Rafael Guzman from Stanford, he's a very good friend of mine. So they did a very excellent study where he injected some of the cells which were nanoparticle tagged, and he found that the 74 cells of per mm² of the infarct had those cells. Well, when he injected 1300, injected through intraarterial route, 1300 cells per mm² was there in the infarct lesion. And this gave a more robust background how we can take this intraarterial delivery superior to intravenous delivery. Now, my lab in the US, Dr. Dilip Yavagal, he was the first interventional neurologist who took the first clinical trial of stem cell therapy IA in the US. He started working, and we started working with him, where we had to prove whether this IV was less or more efficacious in comparison to IA. And our study in 2011 showed that, okay, the intravenous delivery was less efficacious while the intraarterial delivery was more efficacious. And this can be validated for its robustness and can be taken into clinics. But till date, we had many questions before us. So let me take you to the work of Michael Chop of the University of Michigan. What he did, actually, he took three groups. One group was intraarterial, one group was intracerebroventricular, and one group was intravenous. What he did, he labeled the cells with supraparamagnetic iron oxide nanoparticle, and he had the 3D gradient eco-imaging. And if you see, there were the differential time point that he took for the study. It was 4 hour, 1 day, 2 day, 3 day, and 4 day. Now what he did, he conducted an MCO and he injected the cells. Now, if you basically compare with IA-IV at 4 hour, you find the maximum number of IA delivered cells at the stroke foci. But if you compare with the IV group, you only see some of the punctate signals coming out. So this was an evidence that at 4 hour, IA could make it. As I have mentioned, that when you are talking about the intervention of stroke, the time period is very much important, and you have to conquer the narrow time window. So if you are even injecting IV, you do not know whether the cells are going to reach at the defined time, at the less time to the stroke foci or not. And this experiment was continued and it was taken up to 4 days. And even at the 4 day, you find the maximum punctate signals coming from the brain hemispheres in comparison to IV, where you have a single punctate signal. Now we just did an experiment where we had biodistribution cells of IA versus IV, and we checked the proton flux in IA and IV. If you see the IV, most of the cells which were luciferase expressing, they were all at the splenic area. They could not make it to the brain, but if you talk about the intraarterial delivery, that too in the intracarotid, most of the cells reach to the brain. And if you see the proton flux, most of the flux is coming out from the brain region. So this was again a validation that if you are able to deliver the cells through intraarterial route, you can be able to push maximum number of cells to the stroke foci. Now, there were a lot of questions before us. It was not easy to take our research from bench to bedside until and unless you answer some of the questions. So there are a few questions which I just wanted to put before you, because all the researchers before trying to take the research from bench to bedside need to think all these questions. So for the first question, the model that you are going to do, whether it mimics the natural condition or it mimics to the patient, that has to be determined first. What about the concerns of the blood-brain barrier? The cells, when you are pushing those cells to the intraarterial blood circulation. Now, those are microcapillaries. They already have a robust blood flow. If your cells are going over there, do they clog those vessels? And if they do clog those vessels, you are going up to cure stroke. You are coming up with new lacunar strokes. That's not the goal. And how these transplanted cells, when they reach the site of injury, how they reach? Is there any signals this reach to the site of the injury? They also can move to the contralateral portion. Then you cannot say that these cells are moving to the stroke foci and they are very much aligned. You have to have a justification. You have to have a reason for that. Now, you are pushing cells from outside sources. In the patient, it is always autologous. But when we talk about the animal experiments, we always take allogenic cells. So if you are pushing those cells into the brain, how many days they are surviving? Are they sitting in the brain or they are phagocytosed? Because you have microglial cells and they are always doing this cleaning activity in the brain. They may take out the cells and your cells may not do at what has to be done by those cells when they are pushed inside the brain. Now, whenever if you have a stroke, you have a cytokine storm over there. You have acidotoxicity, you have calcium excitotoxicity, you have NMD overexpression. So these are something which are just affecting the microenvironment. Do those cells which are over there, are they able to survive in this adverse condition? The cells that you are pushing, the question is that what the blood is doing with the cells and what the cells are doing with the blood, that has to be noted. And ultimately you come, what is the mechanism of neuroprotection? So it is not easy to take your research from bench to bedside unless and until you answer all those questions. So we have a very successful model where we occlude the middle cerebral artery, and we have a descent infarct volume which can be compared to the infarct volume of humans. So in that case, we try in our lab, we try to do all the conditions that are mimicked in the stroke clinic, whether you are going for a laser Doppler flowmetry, where you're going for brain oxygenation, whether you are going from blood physiological parameters, we are maintaining glucose, we are maintaining temperatures, and many other things. So these things are very much observable, that when we start thinking of taking our research from bench to bedside, whether we are adhering to all those parameters or not. Now we talk about mesenchymal stem cells. So it is not possible to take any cells, even it is MSCs, you need to have some of the criterias. So let me tell you, all the young people who are sitting over there, we had some parameters where we selected these cells. First of all, we took the passage. The cell should not be more than 2 to 4 passage number, because as you move along the more passage number, the genomic instability comes and your cells will start differentiating. We don't want our cells to differentiate. That was the question is. If you are talking about differentiation, if you feel that differentiation is good, then you have to counter one answer that you do not know how many neurons these cells are going to produce over there, or whether these neurons will be functional neuron or not. In that case, you go for treating stroke, but you land up with a glioma, and that is not our intention. We took cells which is having 2 to 4 passage number only to maintain the genomic stability. Now, for my young friends, what is CD45 and what is CD105? This CD45 actually is a transmembrane tyrosine phosphatase. The expression of this CD45 determines the multilineage differentiation capacity, and they give us information whether they are being able to generate fibroblasts or osteoblasts. We want the CD45 to be negative, as we do not want the differentiation. While we talk about CD105 positive, I would mention here the CD105 positive means these cells are prone to differentiation to adipocyte and osteocyte. This means that if you are going to suppress the proliferation of CD44, the CD105, that the endoglin positive demonstrate high trilineage differentiation, means this CD45 shows that you have more or less the control over the differentiation. Then we talk about the viability. The cells, the maximum number of cells should be viable. You are not going to push the debris in the brain. And of course, the size matters. I have mentioned that you have to take care whether your cells are going to cause any microembolic stroke or not. So we fax down cells and make it up to 50, 70 micrometer. And these cells are shorted out and they're taken for infusion. Now, if you talk about the preclinical aspect, the most important, we started with 100 million cells and we de-escalated the dose. And we found that the 100,000 cell was efficacious. I would try to bring your notice over here. When we made a comparative study between IA, IV, MSCs and PBS, we found that IA MSCs at 28 days was improving the neurodeficit score. And even if you talk about the day 28, the long term, the IA MSCs was the most beneficial dose that was giving the maximum recovery in the functional outcome. Now the question, if you are pushing the cells, how you can push the cells? So as I know, whenever there is a stroke, you have more stromal derived factor 1 upregulated in the brain. I take you to the previous slide once. Now, here what we found, that when you are giving IA MSCs as 24 hours, it is showing less benefit. But when you are pushing IA MSCs at one day, it is more efficacious. So what is the reason? You are pushing the same number of cells, but if you are pushing it at 1 hour, it is not efficacious. But if you are pushing at 1 day, it is efficacious. Now we started working on that. So we found that whenever there is a stroke, there is more upregulation of stromal derived factor 1. And when we take different groups and different hours of the expression of SDF1, we found that SDF started expressing more at 6 hours, and that gave an answer to us that at between 0 to 4 hours, there was no expression of SDF1, hence the cells were not homing. Now, how this homing takes place? Let me clear it. There is SDF1 CXCR4 excess. This CXCR4 is within the cells where SDF1 as the infarct area. And there is an attraction, that what we call as a chemo attraction going on. So whenever you have a sufficient amount of SDF1, then only the cells can go to the infarct size. Now let me talk about an experiment where we used as AMD 3100 CXCR4 antagonist. Now we have to prove whether it is only SDF1 CXCR4 or there might be some other reason. To our astonishment, we find those groups which were having AMD 3100, they were not having the maximum number of cells. These were actually the cells which were evaluated from the brain biopsy. We pushed those cells and we took out the brain. And we found those groups which were having the CXCR4 antagonist, they didn't have much MSCs. So this confirmed that anyhow, the stromal derived factor upregulation at a given point of time was responsible for homing of the cells. Now the past evidences often reported that these MSCs are giving neurotropic factors. And the neurotropic factor, if you talk about, it is the BDNF, brain-derived neurotropic factor, which is the most prominent by the cells. And these are more or less more beneficial in the case of stroke. Now the question comes, if it is only the BDNF which MSCs are given, then you can directly give the BDNF to the brain and it can cure your stroke. So no, this BDNF has some challenges. This BDNF cannot cross blood-brain barrier, and you cannot push BDNF directly into the brain. And the second, what the justification we had, that whenever there is a cell and cell communication, the cell knows when to give BDNF and when not to give BDNF. When you're pushing BDNF directly into the brain cannot evaluate what to take and what not to take. So in that case, we wanted a natural cell-to-cell communication, and for that we took MSCs as the main agent which can give a BDNF at a required time, but which can be beneficial for in the stroke outcome. So these are some of the plots where we saw the expressions of two different groups in PBS and MSCs, the upregulation of BDNF and its signaling track B, and we found that the IMSC treatment after stroke increases BDNF and its receptor protein in the penumbra. Now, as I have mentioned the plot before about the SDF, that at 6 hours, you have the maximum expression of stromal derived factor 1. Now you have SDF1 more at 6 hours. You are pushing cells at 6 hours. Now you have to come across whether the blood-brain barrier is allowing your cells to move inside or not. Although people argue that whenever you have a stroke, you have already a compromised blood-brain barrier, but you need to have a rationale on the basis of experiments. Then what we did, we took two groups, we took a sham, we took a stroke, and what we did an Evans blue experiment, and we found at 6 hours, the blood-brain barrier was open and we were able to detect the Evans blue in the pictograph. And if you do a spectrophotometric analysis of the dye, you get at stroke and 6 hours, you get maximum absorbance of the Evans blue dye at 620 nanometer. Now, we have two things in our hand. One, you know the rationale, you know the reason why the cells are going to the injured area. You have the rationale why you are taking the 6 hours as a time, and that 6 hours, you can push the cells across blood-brain barrier. But the question is that you are pushing cells, and those cells are more bigger than the blood corpuscles, whether they are anyhow creating microembolic stroke or not. So for this, we did a comparative blood Doppler, laser blood Doppler flowmetry experiment where we had two groups, the PBS and the MSCs. And I will tell you that we dissolve these MSCs in PBS. So what happened? We tried to optimize or validate the way we inject the MSCs to the brain. So what we do, we take a P10 catheter and we put it in the intracarotid, and the pushing time we optimize, and we found that if we push 100,000 cells in the duration of 10 minutes, this can easily flow with the blood corpuscles and there is no microembolic stroke. So the no deviation in the blood Doppler gives a hint that there is no microembolic stroke in the brain when you are pushing cells, whether it is in PBS, and the pushing cells of MSCs was as equivalent to the as comparison when you're pushing PBS in singlet. So to move forward, we have 3 questions addressed: that you have the blood-brain barrier opened, you have the cells shorted down, you don't have the chances of microembolic stroke while pushing cells. Now you have to see whether the cells are in the brain or not. Before coming to this slide, I would just go to 2 of the videos that was done by me at Boston Children's when I was in Harvard. So these are the real-time imaging of the brain of an animal which were infused with MSCs, GFP positive MSCs. So this is the brain where you have the capillaries, you have a vacuum area, that what we call as the infarct area, where the insult has been made, and you find there is no signals which are coming out from any of the agents that have been pushed. Now, if you see the comparative image over here, just see how small MSCs which are GFP positives are there. So you have the blood-brain barrier open at 6 hours. You have pushed the cells without creating a microembolic stroke. Now these cells are residing at the infarct area, which is being justified by this ultramicroscopy image where you find the GFP positive cells sitting in the infarct area of the brain. Now let me come to the back slide previous. Now to counter confirm, as this was a real imaging, even if you see the image of a single capillary, you find a small cells, and these cells are anyhow moving. If you see the diameter of these MSCs, they do not show any signs that they are able to clog this vessel. Now, to counter confirm it molecular means we did a fluorescent in situ hybridization FISH, where what we did, we took a female animal, we overectomized the animal to negate the estrogen neuroprotection, and then we injected cells, MSCs, from a GFP positive transgenic male. And due to this XY disparity, we were able to locate the signals on the Y chromosome, which clearly mentioned that those cells were inside the brain, and we were able to detect those cells in the brain. And even we went for 2-photon microscopy, and we found that in the period of time when you took an initial scan, you have 7 to 8 number of cells in a given frame, and after 7 to 8 days, you only have 2. So this gives us a hint that cells are always not there. They are sitting over there for some time, they are phagocytosed, but the neuroprotection time window remains for a month. So now what is the reason? So you are trying to address each and every step, each and every question before having a justification of taking it from bench to bedside. Now, as I have asked a question before, that what the cells do with the brain, with the blood, and what the blood do with the cells need to be checked. Now, we did some hematocompatibility studies where we checked that what were the shape of the corpuscles that are being there when you inject the cells. If you see there in the comparison, this is a GIMSA staining of blood smear, you find that there is no difference in the cell morphology if you compare between IA-PBS and IA-MSCs. Even we had many hematocompatibility studies parameter of these 3 groups, and even we checked with the blood with the coagulation factors, including the D-dimer, whether these are triggering any factors which can spoil those cells or not, which can destroy those cells or not. So for our result, we saw that there was no such adverse reaction going on when you are pushing these cells intraarterially, which is coming in contact with the blood. We also did the toxicological study of the animal, and we did a liver function test. We did a kidney function test. We had some H&E staining of different organs, kidney, brain, and lung, liver, spleen, stomach, and heart, and we didn't find any adverse reaction. Now, at the basic bench level, we find that there are no adverse effect of these mesenchymal stem cells when they are given into an animal of stroke. They are not causing any adverse condition. Now, this can be taken further to decipher the mechanism how these MSCs are beneficial. Now, the question comes, whenever you have a stroke, you have a damage, you have cellular dysfunction, and all the cellular suborganelles, more or less, are affected. Now, the major cellular organelle that comes in our mind is the mitochondria, because when you have a stroke, you have a depletion in the mitochondria, ATP, and it's clearly hints that your mitochondrial function is being compromised. Now, for my young friends who want to understand what actually happens, let me give you a brief introduction of this. So whenever you have a stroke, there is an increase in the intracellular calcium level. And now this calcium then moves into the mitochondria by the calcium uniporter present on the mitochondrial membrane. Now, once the calcium concentration inside the mitochondria increases, it leads to the activation of several processes. And one of which is the formation of the MPTP, that what we call as mitochondrial permeability transition pore. Now, this MPTP formation leads to the leakage of essential ions from the mitochondria. Now, when there is a leakage of these ions from the mitochondria, there is a destabilization of the mitochondria. Now, this high calcium concentration now interferes with the ETC, leading to the generation of ROS, which in high concentration actually overwhelms the antioxidant system within the mitochondria. Now, when you have a high ROS, it also disrupts the mitochondrial membrane, leading to release of cytochrome C within the inner mitochondrial space. And this cytochrome C releases the cell towards apoptosis by activating the caspases. Now, you remember mitochondrial membrane disruption also leads to the leakage of ROS into the cytoplasm, and that eventually leading to the damage to the lipids, proteins, and nucleic acids. So this is how the mitochondria is playing a crucial role. Now, we came up with some of the hypotheses which looks very fascinating at initial stages, but we were even not sure whether it is happening or not. We started hypothesizing that when we are injecting these MSCs to the brain, when these MSCs come in contact with the neurons, they may donate their good mitochondria to the neurons. So it looks little bit very fascinating, but we had a clue that whenever there is a cell-cell interaction, there is always being communication between the cells, and cells form tunneling nanotubes between them. Now, if we are pushing MSCs to the brain, when these MSCs come in contact with the neurons, they may be able to transfer their healthy mitochondria to the dysfunction towards the neuron which has a dysfunctional mitochondria. In that way, we can replenish the mitochondria which are not functioning well in the neurons, and that may give us a chance of giving a neuroprotection to the animal or the person in question. It was a big hypothesis. We didn't know what is going to happen. Although we started working on that. First of all, we did a pilot in vitro study. What we did, we cultured the PC12. We have a hypoxia chamber. We put the PC12 cell in a hypoxia. This PC12 cell were hypoxic. And what we did, we co-cultured it with the mesenchymal stem cells. Now, as per our hypothesis, these mesenchymal stem cells should able to give the mitochondria to the PC12 cell cells, and there might be some role of the sirtuins, because the previous studies have shown that sirtuins are one of the drivers of mitochondria. Now, with this, we started working on the in vitro experiment, and it was fascinating for us when we got a very good result. If you see, if you see the picture, if you see the images, you have big MSCs, which are stained with mito-green and which looks very green. And we have small PC12 cells, which are stained with wheat germ agglutinin. Now, if you see, these MSCs have long projections towards the PC12. Now, even if you see the fourth image, you can see small mitochondria are floating through these nanotubes toward the PC12. So initially, we had a good image, which could at least say that our hypothesis may not be wrong. It can have some of the substance, but we need to check it more robustly, how to validate this, how to confirm this. Okay, this only is occurring. For that, what we did a mitochondrial transfer experiment where we took cells. One was the PC12 normoxic cell. We cultured it with the mesenchymal stem cells. One group was PC12 hypoxic cells, and we cultured it with the mesenchymal stem cells. And we checked the mitogreen signals. Now, if you talk about the mitogreen signals, these mitogreen signals only coming from the mitochondria. Now, what we find that those cells which were co-cultured with MSCs were having more mitogreen signals. We didn't stop over there. We started working on the viability, the percent viability. We again took 4 groups. We talk about the normoxic, we talk about the hypoxic, we talk about the co-culturing. So we had 4 groups: PC12 normoxic, PC12 hypoxic, PC12 normoxic plus MSCs, and PC12 hypoxic plus MSCs. And to our astonishment, we found that in the case of PC12 normoxic plus MSCs and PC12 hypoxic plus MSCs, the percent viability varied, and those with MSCs gave a good percent viability. Although this was a vague result, we didn't come to the point where actually what is happening. We had to carry some more experiments. Now again, we went for the ATP consumption study. So what we did after the hypoxia, we cultivated this PC12 alone, and we checked the ATP content in comparison to a normal PC12 cell line. And following co-culture with MSCs, this hypoxic PC12 and healthy PC12 produce the same amount of ATP, indicating that MSCs improve the energetic metabolism of the injured PC12 cells. But when we co-cultured MSCs with the hypoxic PC12, it generated higher amount of ATP than MSCs cultivated with healthy cells. So here the hint came, it likely that there must be some activating factors in the damaged cells which may be activating this MSCs to donate mitochondria towards the cells and giving us increased ATP. Now we went for a qPCR analysis of multiple genes. Multiple genes means we have TFAM, we have CERT, we have, I would like to say over here that this FIS1 or DRP1, these are mitochondrial fission markers. Whereas OPA, MFN, MFN2, these are mitochondrial fusion markers. If you talk about the mitochondrial biogenesis, these are TFAM. And if you are talking about some transfer markers, we will discuss it later. So out of this, the qPCR analysis, we saw that CERT1 was evolving as one of the major contributors. The expression pattern was much more higher in comparison to other genes. So here we found that, okay, CERT1 may be one of the crucial factors which might be driving the process. So let me give you a brief introduction about the sirtuins. What are sirtuins to all my young friends? The sirtuins are basically the NAD plus dependent deacylase having a diverse role in energy metabolism. The sirtuins dysregulation, as per past reports, have a major role in the pathogenesis of a neurological disorder. And this SIRT1 has been shown to be neuroprotective. Now, we are trying to anyhow make a relation with the sirtuin, this brain-derived neurotropic factor, and the inflammasome signaling, which is coming into the play when you have a stroke. So let me tell all my young friends about what are inflammasomes. These inflammasomes are multi-protein receptor-like structures involved in the initiation of inflammatory cascade. And these inflammasomes are one of the major driving players of progression of inflammation and apoptosis. Now, coming to the experiments. Now, what we did, we planned one experiment where we hypothesized that whenever there is a ischemia, you have less sirtuin, and this sirtuin decreases the BDNF, and which enhances the inflammatory mediators and which may take the cells towards apoptosis. And if we administer these mesenchymal stem cells, these mesenchymal stem cells may upregulate the sirtuin-1, which in turn may upregulate the BDNF, and these may able to suppress the inflammatory markers and reduce the process of apoptosis towards neuroprotection. This was our hypothesis, and we started working on that. What we did, we planned experiment where we have ischemia of 90 minutes. Let me tell you, we take 90 minutes ischemia because this mimics the moderate stroke. And we also took one of the sirtuin inhibitors, which are specific sirtuin inhibitors, that what we call as EX527. So what is EX527? It is a selective inhibitor of SIRT1, and it is expected that when you have this inhibitor, if SIRT1 is actually involved in all the process should not go on in that group which has the inhibitor. And then we had a different molecular, biochemical, morphological evaluation to validate our hypothesis. Now, let me talk about the preliminary. What we did, we had 5 groups, and the major group being the stroke plus inhibitor and stroke plus inhibitor plus IA-MSCs. As I have mentioned, we took the inhibitor EX527, where we want to see whether SIRT1 really is playing any role or not. That what was intention. So what we saw, that post IA-MSCs treatment, there was significant reduction in the neurological deficit, and as well as when you talk about the inhibitor, higher neurological deficit were seen in stroke plus inhibitor and stroke plus inhibitor plus IA-MSCs. Even if you talk about the infarct volume, you see whenever you have a stroke plus inhibitor, the infarct volume is more. Now, going into more details, we had checked with the nissls. What we found, that whenever a higher reduction in nissl staining in stroke and stroke plus inhibitor and stroke plus inhibitor IA-MSCs group as well, but there was a reduction in average neuronal density and neuronal length when it was seen in comparison to stroke, stroke plus inhibitor plus IA-MSCs as compared to sham and stroke plus IA-MSCs. So this was somehow bringing us closer to our hypothesis. Okay, what you have hypothesized, that might hold some of the water that can have some of the positive outcome in further experiments. Now we went for some behavior that usually all the pharmacologists do. We took a rotarod, we took a grip strength, and the retention time on the rotarod was significantly reduced in the stroke and stroke plus inhibitor group, as well as stroke plus inhibitor plus IA-MSCs as compared to sham. And what we saw, that following IA-MSCs administration, there was a significant increase in the latency time that was found in stroke plus IA-MSCs group. And there was no difference between these two groups, which were considered as stroke plus inhibitor and stroke plus inhibitor plus IA-MSCs. Now, even if you talk about the grip strength in stroke group, that was significantly decreased as compared to the sham group. Even in IA-MSCs treatment group, there was a significant improvement in grip strength as observed as when you compare with the stroke group. But if you see, there was no improvement in the grip strength of animals of the stroke plus inhibitor and stroke plus inhibitor plus IA-MSCs. We also went for the mRNA expression of SIRT1, BDNF, NF-kappa beta, inflammasome, NLRP1 and NLRP3. So if you see, the levels of SIRT1 and BDNF mRNA were significantly reduced in stroke and stroke plus inhibitor and stroke plus inhibitor plus IA-MSCs groups. But when you treat them with MSCs, this MSCs treatment upregulated the mRNA expression of SIRT1 and BDNF. Conversely, if you talk about the mRNA level of NF-kappa beta, NLRP1 and NLRP3, these were significantly elevated in the stroke and stroke plus inhibitor and inhibitor plus IA-MSCs group. While if you see about the MSCs treatment, they reduce the mRNA expression of these markers in stroke plus stroke IA-MSCs group. We also went for different protein expression studies of SIRT-WIN, BDNF, TRAC-B, TNF-alpha, NF-kappa beta, NLRP1, 3, caspase, ASC, cytochrome, caspase beta, and we found overall that the protein expressions of SIRT1 and BDNF were significantly reduced following stroke, as demonstrated from the result of immunobloating and immunofluorescence. The expression of BDNF receptor as well as TRAC-B was also reduced following stroke. Now, if you check, there was an increase in the expression of SIRT1 and BDNF and as well as the TRAC-B protein when the group had MSC's administration. So even if you say in the same group, MSC treatment reduced the expression of TNF-alpha, NF-kappa beta, and inflammasomes, and also the apoptotic marker cytochrome 3 and caspase 3. So this was anyhow counter confirmation of our gene expression study, and even in the protein level, the same trend was observed. Now, I would like to go for the immunofluorescence study of the same, where we had the same kind of result. If you talk about the SIRT1, even if you see that the expression of the SIRT1 in the IMSS group was significant. If you talk about, I'm not taking much time on this. So if you talk about the BDNF, the same trend as we've got in the protein expression, we found that stroke plus IMSCs had the maximum expression of BDNF. If you talk about the immunofluorescence of NF-kappa beta, you find that stroke plus IMSCs had the reduced NF-kappa beta expression. If you talk about the expression of NLRP1, even in the same trend is there, stroke plus IMSC group had the lesser amount of NLRP1, NLRP3. Now, if you talk about the tunnel positive cells, even if you see that there was a robust expression of tunnel positive cells in the stroke group as compared to sham group. But when you observe the tunnel positive cells in stroke plus inhibitor and stroke plus inhibitor plus IMSCs, the number of tunnel positive cells was significantly reduced in stroke plus IMSCs group. So this was anyhow the counter confirmation what we are doing, that SIRT1 was more or less involved in driving the process. Gaurav, do I have time?
Yes, sir. Another key speaker is also waiting, sir.
Can I take five more minutes?
Yes, sir. You can, sir.
Yeah. So let me just there are more data. So okay. So I should just come to last. There are many studies. Let me come finally how we are taking it from bench to bedside. There is a canine model of stroke that what we are doing. So I was well trained in Harvard to doing a stroke surgery in the dogs. And we started putting a coil at the dog M1 terminal, and we were able to produce a big infarct in the dog brain. And the MRI flare clearly mentions that when you are able to lodge this coil at M1, you have a big infarct. So now we administered 100 million cells to the dog intraarterial, and we found, if you see the MRI, you see the infarct is gone. So this is something which was very promising and convincing for us. We again went for a subacute administration of 100,000 million cells to the dog. And even if you see the result post-stroke, 30 days at flare, you see the infarct volume has reduced. We also did some more experiments with the number of animals, and we had the same result. Even with the treatment of IV MSCs, we took an ex vivo study where we did an IV MSC comparative study, and even in the IV MSCs, we didn't find any such improvement. Now, we also went for diffusion tensor imaging, where we were able to track down the nerve fibers coming out of the brain, and we saw the improvement in the imaging, the fibers, which were better in comparison to that you had before the stroke and when you administered the canine with the MSCs. So these are some of the large animal study. I am not going to take much more time. Thank you very much. First of my collaborators, my boss at US, Dilip Yavagal, Nobel laureate Andrew Shelley. He's still 91 and he's collaborating with me in many aspects. And Zain from Brigham, and my PhD students. I have to include two more. I have 8 PhDs now. MS students, junk of students. And this was my house in the US, just beside the lake. And my wife used to take a cup of tea in the evening when I come exhausted, and that refreshed us. Thank you very much.
Thank you so much, sir, for this enlightened talk and elaborated talk on the stroke and their pathophysiology, their neuroprotection. So actually, I am also your junior and working on the same, not such an advanced. So thank you so much, sir, for joining us and this enlightening, motivated talk. I would like to request Professor Renna Patnaik, ma'am, to kindly facilitate our chief guest with memento of honor.
Thank you very much, Dr. Pallav, for such an informative lecture, and very nice to see that you have done such work. And now, on behalf of Galgotias University, I request you to accept the e-memento.
Thank you very much, ma'am.
Thank you, sir. Thank you so much, sir. Thank you once again, sir. Now, dear participant, we are moving for our next session, and we have another eminent speaker with us, Dr. Santosh Kumar, sir. Hello, sir. Dr.
Dr. Gaurav, what about the e-memento? We have not.
We have given, no, ma'am?
No.
Yes, we have shared.
You have shared. Okay, fine.
Yes. Okay, so I would like to introduce our speaker, Dr. Santosh, sir. Dr. Santosh Kumar sir is currently working as research professor in Division of Chemical Engineering, Konkuk University, Seoul, South Korea. He worked as a research associate at Department of Chemistry, MNIT, Allahabad. He was awarded CAS TWAS postdoctoral fellowship in 2008 and also worked in Changzhou Institute of Applied Chemistry, China. Later, he joined as a Konkuk University brain pool professor and research at the Konkuk University, Seoul. He was awarded FCT postdoctoral fellowship in 2012 and worked in Chemistry Center, University of Coimbra, Portugal. And during his career, Dr. Kumar has published a more than 90 plus research paper in SCI Index Journal with high valued journal and attended and participated in local and international workshops and conferences. He also served as a member of different organizing committee and an editorial board member and reviewer at number of scientific journals. Besides, he is also associate editor of Asian Chitin Journal and editor-in-chief of Journal of Biological Engineering Research and Review. He is member of Royal Society of Chemistry, UK, and Indian Chemical Society. His current research interest involves biomaterial, nanobiotechnology, nanotechnology, biomedical, and environmental application. So I would like to request Professor Renna Patnaik ma'am to kindly invite our keynote speaker, Dr. Santosh sir. Okay. Okay.
Welcome, Dr. Santosh, and I request you to present your lecture. And so nice that you have accepted our invitation on this National Science Day. So please present your matter.
Good afternoon, ma'am, and all of you. So first, I would like to thank organizer and Chancellor and Vice-Chancellor of Galgotias University, who has invited me on my talk on the occasion of National Science Day. So I'm going to share my slide. So slide is visible, Gaurav?
Yes, sir. Yes, sir. It is visible, sir.
So today I'm going to present our work about porphyrin, a versatile smart material. The slide is visible.
Yes, sir. Your slide is visible, sir. Do it in full mode. You can play the slide.
Yeah, full mode is.
Yes, sir.
So you can see this all the structure about this is BMW car, and this
Sir, the slide is not moving.
slide is not moving.
Yes, sir.
1 minute.
Yes, now it is moving, sir.
Okay, so I will not do full mode. Is it moving?
Yes, sir. Yes, sir. Yes, sir.
So if you can see, what do all things have a common? So this is a car, and this is the photodynamic therapy, and this is the blood, and this is the vegetable chlorophyll, and this is the meat, red meat, and these are the solar cell, photovoltaic solar cell, and these all involve directly or indirectly a class of chemical compound known as porphyrin. So first I want to show about the history of porphyrin. So porphyrin began with dye extracted from indigo plant. They were so brilliantly colored and color fast that they quickly became a luxury royal purple. So a new German company, BSF, began manufacturing synthetic indigo dye. And after that, a company ICI, England, trying to make a modified indigo, kept getting a purple byproduct that contaminated their desired product. So impurity were also brilliantly colored dye, which could withstand temperature up to 500 degrees centigrade, and other dye are is not stable than a 500 degrees centigrade. So this is a very good dye for color fastness. It's also showing very color fastness. So we have also one history, Champaran Satyagraha movement led by Mahatma Gandhi in India, and that time he was just increase farmers uprising, indigo plant farmer during the British colonial period. So why porphyrin are cool? Because they are really big. They can contain many different metal atoms in their center, because they are very stable due to their conjugation. Because if you see in next slide, porphyrin have a alternating single and double bond, because they can take on an amazing variety of role in the real world, which is why they are widely utilized and researched. So if you see a structure of porphyrin, so this is the parent porphyrin, and here is a 4 pyrrole ring, and it's conjugated with 4 methane bridge. This parent structure we called porphyrin, and when if you see here substituted porphyrin, so that porphyrin that substituted porphyrins we called porphins. So here we can modify many porphyrin. So, and complex, we have already synthesized tetraphenyl porphyrin and many other porphyrin and methylated tetraphenyl phenyl porphyrin and other porphyrin. And we already published one review chapter in a review paper in a title about covalently linked free base and metallo-base porphyrin, chemistry and diversity. It published in Current Organic Chemistry in 2015. So use of porphyrin. So we know because of the unique chemical structure of porphyrin, and it can use as a metal binder ligand, as a solar cell. Porphyrin can absorb a photon from the sun and convert into electricity or chemical energy. It can also use as a oxygen transport medium in our hemoglobin in blood, as an electron transfer medium conducting polymer. It has also very useful in hormone synthesis, in gene regulation and gene vector, and drug metabolism, iron metabolism, and also environmental application like CO2 capture and conversion. So here, if you see this the heme natural occurring porphyrin, here iron is containing in the central metal in center in porphyrin. So this the porphyrin structure. So heme, that is the also it's a Greek word hema, meaning blood. So if you see in chlorophyll used as a photoreceptor. Here you can see chlorophyll structure, magnesium in the center of porphyrin, and the difference between heme is iron atom in hemoglobin, and in chlorophyll magnesium. So in porphyrin is contain chlorophyll and hemoglobin also. So in also used as a mitochondrial electron transport chain. So in the cytochrome C, if you see here, so a molecule responsible for transporting an electron used to provide energy to the organism. And these molecules are identical for related species of plant or animal. So for example, chicken and pigs have identical cytochrome C. So if you see myoglobin is a muscle cell, it's also responsible for color of red meat. So here you can see this chicken meat have a very less myoglobin, and if you see in red meat, it's a lot of myoglobin. That's its color is also showing red due to porphyrin. So and vitamin B12, you can see vitamin B12 here. Cobalt is the central atom. That's why it's called as cobalamin. Also, it's also directly linked with porphyrin. So, and water splitting molecule in fuel cell, they produce hydrogen gas from water. So porphyrin based photovoltaic are used directly for power need. So porphyrin here can absorb store chemical that can store a photon and is changed in during water splitting hydrogen and oxygen. So chemically storage is a more viable option compared to battery in terms of efficiency and weight. So and the stored hydrogen and oxygen is fed into the fuel cell at night for continued use. So in nanomedicine also porphyrin can is a very big role. If you see porphyrin methylated methylated porphyrin and linked with lipid polymer drug peptide at self assemble. So you can get a liposome misses nano assembly polymeric nanoparticle. So that can be used in MRI, SPCT, PT, NIRF, PAI, PDT, PTT. So nanomedicine improved bioavailability, better pharmacokinetics. It's a targeted therapy and multifunctionality. So for various forms of nanomedicine have been developed, which include liposome, micelles, nano assemblies, and polymeric nanoparticles that have already achieved remarkable success in developing cancer therapy. So our chief guest already showed many things about nanomedicine. So nanotechnology can be used to improve bioavailability, selectivity, and activity of porphyrin. So for numerous type of porphyrin-based nanomedicine have been established with superior anti-cancer therapeutic effect and excellent imaging properties. So you can see photodynamic therapy. So injection, so drug can injected by injection. This is the tumor cell. So that drugs, porphyrin related drug can accumulate as a drug in tumor cell, selective, and irradiation of drug with light because have a porphyrin structure. So, and you can see in 4 stages tumor necrosis. So, and the conducting polymer also, if you see our, if we can use conducting polymer and so, that photovoltaic coating or our house top roof of house. So that then you can use power your house or a vehicle. If your vehicle was painted with a photovoltaic coating and a road also, so in collect so that photovoltaic coating can collect sun light and emit the electricity needed to power itself. This would be environmentally friendly and in most cases cheaper than the petroleum product. So if you see photovoltaic polymeric photovoltaic coating with cloth, so from this we can use our we can recharge our mobile laptop. That's porphyrin which are chemically similar to chlorophyll. So that can also chlorophyll used as a photosynthesis. So and that porphyrin can use as a artificial photosynthesis. So can generate energy. So we have also worked on porphyrin as a nanoreactor in the carbon dioxide capture and conversion. We published this paper in Journal Material Chemistry in 2015. So we have converted epoxide to cyclic carbonate, formic acid, and carbon monoxide. So porphyrin research. So our group synthesized a novel porphyrin for a biomedical application and environmental application. So we synthesized a water soluble tetra 4-carboxyphenyl porphyrin grafted ketogen. So ketogen is a natural product, natural biopolymer. It has a copolymer of N-glucosamine and N-acetyl glucosamine. So ketogen is a abundant biopolymer after cellulose. So we modified with porphyrin. So our collaborator, they have worked on our material about porphyrin grafted ketogen. So you can see a different cell line, F549 HeLa cell, HEP-G2 cell, IEC6 cell, L929. And so if you see here, this is the this is a commercially available gene vector PI25K lipofectamine. And so our material, it's enhanced the cell viability in all the cell lines. And also transfection efficiency, if you see here, we have also here you can see a chitogen DNA and our chitogen porphyrin DNA complex. So here, and we used also lipofectamine DNA and P225K DNA, which is available commercially. So in all the cell, you can see in HeLa cell, HEPG2 cell, and IEC6 cell, L929, LN929. It's also showing enhanced in vitro transfection efficiency. So that material is very useful as a non-viral gene vector. So we have already one published in Journal Material Chemistry B in 2013 about DNA TPSC and DNA chitogen complex in a. This is the mechanism. So we already published this work in JMC B. So and chitogen grafted porphyrin, if you see at fluorescent intensity at 433 nanometer of excitation wavelength. So we got a 680 nanometer at a emission wavelength. So that can be used in biosensor and biomedical and also photodynamic therapy. And some also we have modified TPPS with chitogen, and we have enhanced carbon dioxide capture and conversion epoxide to cyclic carbonate. So this work is already published in Carbohydrate Polymer in 2017. So here if you see UV visible spectra, because this is the Q solid band of TPPS, and this is the Q band. So when this one red line is the chitogen derivative TPPS. So here we can see, because why its intensity is decreased, because of J aggregate. Because in the if you see there is a two type of aggregation, H type aggregation and J type aggregation. In H type aggregation can work in a neutral or a weakly acidic condition, and J type aggregate at a more acidic condition less than pH 2.5 or 2.5. And also the aggregation behavior depend on the ratio of porphyrin to chitogen. So this is the CO2 capture of chitogen TPPS. So you can see in this figure, this one is the TPPS, and this one is the chitogen. Chitogen have a no CO2 adsorption, and this the TPPS have a little bit 0.2 millimole carbon dioxide per gram. But in you can see in the case of CH-TPPS, our derivative have a 0.9 millimole CO2 per gram compound. This work is already published in Carbohydrate Polymer in 2017. So we also we have also convert CO2 CO2 with epoxide to cyclic carbonate. We analyzed by proton NMR. So here you can see that is the epoxide ring, epoxide peak, and here this one is a cyclic carbonate peak. So we can just we can just use intensity, and then we can say about conversion yield. So this work is published in Carbohydrate Polymer. So and also we have modified graphene oxide because we synthesized graphene oxide from graphite and modified with thionyl chloride, here chlorine, and that chlorine we used amino TPP, so modified, and we have this graphene oxide cobalt ATPP, that that material can work as a artificial photosynthesis, and we convert formic acid. This work is be published in Journal of CO2 Utilization in 2018. So this is the proton NMR of amino TPP and mass amino porphyrin. So this is the same image of graphene oxide and graphene oxide cobalted porphyrin. So you can see highly porous and have a high degree of macro porosity. And if you see at photocatalytic activity of graphene oxide have it's very zero, negligible. And if you see pure amino porphyrin have a 9%, but and cobalted ATP have 12.03%. But when our compound graphene oxide cobalted ATP have 48.53% NADH photo regeneration. So here you can see in that material also used as a artificial photosynthesis of formic acid from carbon dioxide. So we can see here 96.49 micromole we have convert at 2 hours. So this is the mechanism. I'm not going to detail about mechanism. If you see, that is the porphyrin grafted graphene oxide. So porphyrin can take photon energy, and that electron transfer, and that this graphene oxide transfer electron to MB, and that and it's also changed in NAD to NADH, and with format a dehydrogenase enzyme and carbon dioxide to convert to formic acid. And we also used a sacrificial electron donor, TEOA. So this is the synthesis of porphyrin basic. So you can synthesize by use about aldehyde and pyrrole, propanic acid, acetic anhydride. So you can get this compound. This is the known reaction. About reaction name is Adler-Long reaction. We also worked on conversion of CO2 by cycloaddition of CO2 to propylene oxide using meso substituted free base and cobalt metalloporphyrin as a catalyst. So because we know porphyrin porphyrin internal surface area is high, very 5,000 meter square per gram, and high void volume 55 to 90%, and low density from 0.21 gram per centimeter. So that is are not available in other catalysts. So here we synthesized porphyrin. You can see this is the different substitution about bromo, chloro, and CS3. So here you can see we synthesized porphyrin. That porphyrin chloro TPP, cobalted chloro TPP have a 98% conversion, and bromo TPP cobalted bromo TPP have also 98% from the other TPP. So we calculated a conversion about a proton NMR and 13 carbon NMR. You can see this cyclic carbonate, 13 carbon NMR. These are we have calculated a ton and TOF for a TPP and a bromo TPP. So if you see here, this is the bromo TPP ton, and this the pure TPP. So you can see it's 100, but here it's a high 500, and about you can see bromo TPP. Its top is 96, and pure TPP have a top is very less. So this is the mechanism of cycloaddition of reaction. So here you can see and that is the epoxide, and this is the cobalt methylated porphyrin. So here oxygen atom of the epoxide interact with the cobalt methylated porphyrin, and nucleophile. This is the nucleophile iodide ion of co-catalyst. We used NBU4Ni. So attack on the less hindered. Here is the less hindered side of epoxide, and it's open the ring, and after that carbon dioxide react with the intermediate to form a metal carbonate species. Here it's a intermediate. So and after the intramolecular SN2 cyclization, iodine is liberated, and we can get a cyclic carbonate. So here you can see from our result, chloro TPP and bromo TPP cobaltate were found to be highly efficient catalyst with the highest turn and top for the reaction. So a conclusion is what is there is to research. So that there are a lot of variation. We can change a lot of variation in porphyrin, and lot of substituted porphyrin. When you change the group attached to it any other group and metal, so you can get a new characteristic. So thank you. Any question?
Okay, thank you so much, sir, for the informative talk.
Thank you.
Yeah. And for accepting our kind invitation to join this occasion. So I would like to facilitate our speaker. Kindly accept our gratitude of honor, a small token of love from the Galgotias University, Institute Innovation Council, from the Institute Innovation Council of Galgotias University. So kindly accept our moment of honor, sir. Thank you so much, sir. So dear participant, we are moving for the next session, that is poster presentation. Actually, we have received 25 entries. So you can unmute yourself. I am giving you I am allowing you to all to unmute yourself, and there was a 25 poster we have received. Out of that, we have shortlisted 15 poster for the presentation. That is Chama Misra from BTech student is there. Aditya Kumar. Aditya Kumar, not there. Okay. Chama Misra, are you there? Are you here?
Yes, sir. Good afternoon.
Okay, good afternoon. Aditya Kumar, BTech CS. Not. Anamika Rajvanshi.
Yes.
Okay. Sumit Kumar.
Yes, sir.
David Samuel? Samuel? No. Dr. Rajesh Yadav? No. Disha Gupta?
Yes, sir, present.
Okay. Pallavi? Miss Pallavi? Miss Pallavi, MSc Medical Biotechnology? Anisa Tiwari?
सर मैं प्रेजेंट हूँ डॉक्टर राजेश यादव सर.
Okay, okay. Thank you, sir. Thank you.
Okay.
Okay, sir. Miss Pallavi, Anisa Tiwari, BTech Computer Science. Anisa. Siddharth Singh. Okay. Shweta Mishra.
Yes, sir, I'm here.
Okay, Siddharth is not present. Now Shweta Mishra. Subra Jyoti.
Yes, sir, I'm present.
Okay. Neha Sethi.
Yes, sir, present.
Okay. Ayushi Panchal. Okay. Now only 1, 2, 3, 4, 5, 6, 7, 8. So 8 participant is present here.
Sandeep Srivastava also.
Who?
Sir, Sandeep Srivastava.
Sandeep Srivastava, but we have not received your poster.
Okay.
Srivastava. Sandeep. No? Okay, so Sandeep, you can present your poster at the last after presentation, after completion of the presentation of this student. So you send me the detail. Please note my number and send your name, name of institute, program, semester. Then I will enter your name in our sheet. So kindly send your details.
Okay, sir.
Okay. Now, so make sure that all participants whose name has been announced are co-host or not, because the co-host can only share the screen. So who I am asking for the name again. The Chama Mishra.
Yes, sir. Good afternoon.
Okay, good afternoon. You can share your screen.
Okay, sir.
Please share your screen.
Yes, sir.
Okay. Okay. Sorry to interrupt you. We have Mr. Ashish Rai with us. Ashish.
Yes, sir. I am
Yes,
present.
okay. So Mr. Ashish is my colleague and he is an emerging researcher in the field of biotechnology. He will be serve as an evaluator for this poster session. So welcome, Mr. Ashish.
Thanks a lot, sir, for giving me such a nice opportunities.
Thank you, dear. Okay, Chama, start your presentation.
Ma'am, go.
Each and every participant have only 5 minutes. So take your time, use your time very wisely.
Yes, sir. Good afternoon. My name is Chama Mishra from BTech second year. सर हमारी वॉइस क्लियर हो रही है?
Yes, you are audible.
sir, to we represent our poster on the basis of environment. If we introduce the National Science Day, we remember the discovery of Raman effect. So we celebrate the National Science Day. Then our poster अर्थ बेस्ड, जो हमारे मतलब इस इंडिया या जो है हमारी कंट्री में यूज किए जा रहे हैं, उनको हम जो है सही तरीके से संरक्षित करके अपने पर्यावरण को बचा सकते हैं। जैसे कि हम देखते हैं कि हमारे जो फूड पार्टिकल होते हैं, अनवेस्ट मटेरियल होते हैं, हम उनका डस्टबिन भी यूज कर सकते हैं। अदरवाइज हमारे जो फैक्ट्रीज के जो है वो कार्बन डाइऑक्साइड वगैरह जो बाहर निकलती हैं, हमको उनको रिसाइकलिंग करके फिर से यूज कर सकते हैं। इसी अदर जैसे हमारे इंडिया कंट्री में विभिन्न जो है डिफरेंट टाइप के
Your voice is not coming, Chama. Your voice is not coming. Okay, Shama has lost her connection. Next presentation, Anamika Rajvanshi. Anamika. Anamika.
Yes, sir.
Yes. I have made you the co-host.
Yes.
You can share your screen. If you are not able to share, then I will share your screen. So please let me know. Anamika. Anamika, your voice is not coming. Yes. Anamika, your voice is cracking. Okay. The next candidate is Sumit Kumar. Sumit Kumar.
Yes, sir. Good afternoon, sir.
Good afternoon. Sumit, you can share your screen.
Okay, sir.
And after the poster session, we have a short quiz that will have 25 questions, MCQ, and we will give 5 minutes to attempt that MCQ, and we will announce the winner names in the last of the session after completion of the quiz. So you can motivate your other colleague to participate in the quiz also by sharing the link.
Hello.
Okay.
Hello.
Yes, yes.
Am I audible, sir?
Yes, you are audible.
Good afternoon, sir, and my respected teachers, and all my friends. Today I am here to present my poster on the National Science Day. जैसा हम जानते हैं कि नेशनल साइंस डे हमारा 28 फरवरी को मनाया जाता है, और ये डिस्कवर्ड करा था हमारे सीवी रमन सर थे, उन्हें डिस्कवर्ड करता है। इनका पूरा नाम है चंद्रशेखर वेंकटा रमन, और इनने इसमें करा क्या था कि इनने रमन इफेक्ट को डिस्कवर्ड करा था। इसमें क्या करा था इनने, स्कैटरिंग ऑफ लाइट, एक प्रिज्म लिया, प्रिज्म में से एक व्हाइट लाइट पास करी, उसमें से 7 कलर्स स्कैटर्ड हुए, तो उनने इस इफेक्ट का नाम अपने नाम पर ही दे दिया, रमन इफेक्ट। इन्हें फर्स्ट नोबेल प्राइज इन्हें जीता 1930 में फिजिक्स में, और इनके साथ जो इनके को डिस्कवर थे, जो इनके साथ डिस्कवरी करते थे, तो उनका नाम था KS कृष्णन। और
कंप्लीटेड।
और सर इनके एक अपना जर्नल भी इन्हें दिया था, नेशनल जर्नल ऑफ फिजिक्स, ये दिया था 1926 में दिया था। और इनने एक अपनी एकेडमी भी एस्टेब्लिश करी थी, जिसका नाम दिया था इन्हें इंडियन एकेडमी ऑफ साइंस। और इनकी डेथ 21 नवंबर 1970 में हुई थी बेंगलुरु में। यस सर, कंप्लीटेड।
Okay, thank you.
थैंक यू सर।
Next candidate is for the poster presentation, Dr. Rajesh Yadav, sir.
Thank you, sir.
Sir, you are you co-host or
I'm
not?
sharing screen.
Sir, are you co-host or
I
not?
think
Okay.
so
I
no,
have made
no
you the
co-host, sir.
co-host. You can share.
So, but
Yes, sir. Yes, sir. You can share.
yeah, I can share the screen, sir.
Yes, sir.
Okay. Okay, sir, this is visible. Is it visible, sir?
Not yet, sir.
Hello.
No, not yet, sir. Yes, it is visible.
Okay.
Yes, sir.
Okay, visible, sir.
Yes, sir. Yes, sir.
Okay. Good morning, everyone. This is Dr. Rajesh Yadav from Life Science Division, Department of Bioscience. We will discuss about the role of the aerosol on environmental cooling. So I have made this poster on the environment related. So how can the aerosol affect the environment? So it can be affected by 2 types. One is a direct effect and another is an indirect effect. So how is direct effect? If the aerosol absorb the sunlight, it will increase the temperature of the environment. Or if it scatter the light, then it will decrease the temperature of environment. So this is the direct effect. And one of the experiment also has been shown in the graph here. So different type of the radiative forces present in the environment or aerosol may have different type of the particle. For example, it may contain black carbon, which is released by the fuel, or primary organic aerosol or secondary organic aerosol, BB, that is a biomass burning. So these all can affect, either they may scatter the sunlight or they can absorb the sunlight. So in this way, they will affect the environment. So it has been observed that among all these radiative forces, this red one, that is sulfate, it more scatter the sunlight, while the black and white carbon that absorb the more sunlight. So the result is either cooling or either decreasing the temperature. But it has been observed that include the all forces, it has been observed that most of the scattering occurs by the sulfate and absorption is very less. That means the conclusion is that these all forces are combinedly decrease the temperature of the environment. That means it is responsible for the cooling. So this is the direct effect. Another is the indirect effect. So how it affect indirectly, we can understand by this slide. Here it is shown that cloud information. That means aerosol is responsible for the cloud information. So the size of the aerosol 0.2 micrometer, these are combined with the 100 molecules of the droplets or 100 aerosol and form the 20 micrometer size of the clouds. So in this way, if the size of the droplet will increase, the size of the cloud will also increase. And in this way, the height of the cloud will also increase. But they settle very slowly. So that is why the time will take more. So this phenomena is called cloud albedo. Cloud albedo is that the reflection of the sunlight. So if the large number of the aerosol is present in this cloud, they will scatter large number of the sunlight, and this phenomena is called cloud albedo. So cloud albedo can be measured either 0 or 1. That means 0 means no scattering or 1 means 100% scattering. So it has been shown that if in the there is another effect is called ice nucleation. So aerosol cause the ice nucleation. So as the droplet will increase in the cloud, so size of the cloud will increase. But what will happen if the aerosol have black carbon, or as in the figure is shown, the aeroplane is releasing the fuel. So this fuel will increase in the cloud and it will precipitate the water. That means it is responsible for the dehydration of the water. So as a result, the raining will decrease and precipitation will occur. So that means if the black carbon present in the aerosol, it will increase the temperature of the cloud, and in this way it will increase the temperature of the environment. So these, in this way, these affect. So it has been observed all these things, either decreasing the temperature or increasing the temperature, but greenhouse effect basically increasing the temperature. So conclusion is that of the poster, the aerosol is decreasing the temperature of the planet, but instead of greenhouse is increasing the temperature. So aerosol cooling effect cannot be compensated by the greenhouse effect. So the conclusion of the poster is that aerosol is responsible for the cooling of the planet. So in order to remove all this enhancing the temperature of the environment, different type of the treaty has been observed, which is shown in the figure here, like Kyoto Protocol and Paris Agreement. This has been shown, and as the solution, it has been shown that electric tram is going on. That means it will responsible to reduce the aerosol and decrease the temperature of the. Okay, so thank you very much. This is the conclusion. Thank you, Gaurav.
रजिस्ट्रार से बात करो, वो प्रोफेसर से बात करोगे तो मुझे लगता है कि वो करेंगे। फिर वो फिर बात गई करने।
Thank you so much.
रजिस्ट्रार बोला कि हम
Very
करवा
nice
देंगे।
presentation. The next candidate is Disha Gupta.
Yes, sir. I'm audible, sir.
Yes, you are audible. Are you co-host or not, Disha Gupta?
Sir, yes, I am.
Okay, then you can share your screen. And dear participant, please be ready with your poster. Disha, are you facing any difficulty?
Yes, sir. I'm not able to share my screen, sir.
Okay, so I will share your screen. Just give me a moment. I am finding your presentation actually. This why not this your co-host, but okay. So now you can explain your slide.
Okay, thank you so much, sir, for the presentation as I'm facing some problem. So first of all, I'll be starting with very good afternoon to all the respective teachers and the eminent speakers and all the students present here. I am Disha Gupta, student of BSc Nursing third year, going to represent my poster on the occasion of National Science Day. But first of all, let me say that God must bless the soul of Dr. C.V. Raman, who has given us the Raman effect. And on this auspicious day, we are celebrating National Science Day. The theme which was given for 2022 is Integrated Approach of Science and Technology for the Sustainable Future. In this poster, I have explained, and by keeping this in mind, I have prepared this poster that technology plus science leading to the sustainable development of our future. I have shown an industry that is a brick industry, or there can be another industry such as asbestos industries, silicon industries, sugarcane industries, cotton industries, which release very much amount of smoke in the environment. And the people, those who are working in such kind of industries, are suffering from the major kind of respiratory diseases and related to the circulatory as well as the cardiac diseases. The person, those who are working in these industries are the people, those who suffer from, for example, the diseases can be asbestosis, silicosis, bagasosis, pneumoconiosis, and other respiratory diseases, which will be further moving towards the cardiac problem. Now, when we are talking about the sustainable development, what is sustainable development? It is basically the development or the restoring the resources for the use for in the future, which means the people, those who are using the resources, should use the resources in that much amount that they can be safe for the future generation as well. We are using an enormous amount of coal, petroleum. Do you really think as increasing in the population, we will able to store these kind of resources for our future generation? No. So let me introduce that there should be a sustainable development in our country or all over the globe with the integration of science and technologies. And if we talk about the industrialization and the urbanization, the involvement of technology should be in such a manner that means there could be the implantation of the chimneys in such areas which will be leading to the less disease condition and saving for the more people. And the smoke which is released by such kind of industry should be purified. And if these kinds of pollution or this kind of air is going in out of the environment, the people, those who are inhaling, should not be suffering from any kind of disease condition. If we save the human will save the environment. Next, moving towards the volcanic eruption. The volcano eruption, the major was occurred in the Hiroshima in the Japan, which leads to the many major kind of congenital abnormalities seen in the children. Till present date, the children, those who are born on that major land, are seeing the congenital abnormalities such as Down syndromes, neural tube defect, Klinefelter syndrome, Turner syndrome, superfemale syndromes. These are such kinds of syndromes or abnormality which occur because of either deletion of the chromosome or the duplication of the chromosome. But what is the leading cause? What is the leading etiological factor behind this? Is that using of the environment in the exaggerated manner, using of the resources in the exaggerating manners leading to the volcanic eruption. Here, next, I have shown a beautiful picture of a sustainable development, which means storing our environment in such a way, storing our beautiful resources, storing our Mother Earth in such a way that it could be used and it could be possibly available for the people, for the next generation, those who are upcoming. So here I conclude my poster by saying that science is an integrated form of technology plus biological livings. We need to restore it for the future development. Thank you.
Thank you. Thank you so much for the nice presentation. Yes. The next participant is Sweta Misra.
Yes, sir. Good afternoon. I'm sharing my screen.
Yes.
Good afternoon, sir, and a very good afternoon to all the participants. I'm Shweta Mishra. I'm presenting my poster here. And on the occasion of National Science Day, we celebrate on 28th of Feb on the remarkable discovery done by Dr. C.V. Raman's, very well known as Raman's effect. And here I'm denoting the few discoveries and recent achievements we have done by our nation. I have denoted like Chandrayaan, the aerospace discoveries, achievements we are doing, and telecommunication, even medical sciences, as we have been facing. We are in the war zone of COVID pandemic, COVID-19, and our scientists' hard work and researchers' hard work has given us the strength of fighting with the pandemic, and we are on the borderline of winning this war. And I have shown here few achievements in the field of defense also. We have our own missiles, fighter jets. There are so many startups done by DRDO also. They are also supporting, and we have been working on that. So I believe that we will be fulfilling the vision of Dr. C.V. Raman's to invent or to have new discoveries and to invent new branches of science and achievements. Thank you. That's my topic. Thank you.
Thank you so much for completion this presentation in the stipulated time. The next candidate is Subhra Jyoti.
Yes, I am present here.
Okay.
I'm sharing my screen.
Yes.
Sir, my screen is visible.
Yes.
Good afternoon, everyone. I am Subhra Jyoti from Galgotias University. I am a student of BSc Medical Biotechnology, and the eagerness to understand the biotechnology makes me to choose my presentation topic, that is biotechnology, the science of the future. Biotechnology is the technology which can recreate the nature more beautifully. It is the use of biological systems found in organisms or the use of the living organisms themselves to make technological advances and adapt those technologies to various fields. These include applications in multiple fields, from agricultural practice to the medical sector. Colors of biotechnology. While different branches or divisions of biotechnology are most commonly referred to using colors, there are basically 10 divisions, but sometimes it is hard to define their borders. The most prominent ones include red, yellow, and green biotechnology, but the others are also very interesting as well. I will try to explain them one by one to get better perspective on the matter. First is red biotechnology. It refers health, medical, and diagnostic sector. Second, white biotechnology. It refers gene-based bioindustries. Third is yellow biotechnology. It refers food and nutritional science. Fourth, gold biotechnology. It refers bioinformatics and nanotechnology. Fifth is green biotechnology. It refers the agricultural and environmental biotechnology. Sixth is brown biotechnology. It refers arid zone and desert areas. 7 is blue biotech. It refers aquaculture, coastal and marine areas. 8 is purple biotech. It refers patent, publication and inventions as well. 9th is gray biotechnology. It refers classical fermentation and bioprocess technology. And the last one is dark biotechnology. It refers bioterrorism, biocrimes, biowarfare, and anti-crop warfare. Now, I would like to finish my words and let us go out and create more opportunities for biotechnology and also for our country. Thank you everyone for listening to me.
Okay, thank you so much, Subra, for the nice presentation. The next candidate is Neha Sethi. Neha, are you here?
Yes, sir. Yes, sir.
Okay.
So I'm sharing my poster.
Yes, please.
So is it shared?
Yes, your screen is visible.
Yeah, thank you, sir. A very good afternoon to one and all present here. Myself Neha Sethi from BSc Nursing second year. Today, I got the opportunity to present my poster on Science Day. So I want to include, Science Day is celebrated on 20th February every year. We celebrate National Science Day to remember the discovery of Mr. C.V. Raman effect, and this discovery also earned him the Nobel Prize in 1930 in the field of physics. So in Science for Asia, the day is of great importance for Indian science and scientific. The Raman effect was the wavelength of a small friction of the radiation scattered by certain molecules differs from that of the incident beam. So in this poster, I want to present our India launched ISRO in this space. ISRO means Indian Space Research Organization, and their headquarters are located in Bengaluru. The goal is to develop space exploration for national growth by researching space science and planet exploration. And in this, the boy in the poster is waving the Indian flag when ISRO is launching in this space. And it is a very proud moment for every Indian that we achieved our goal and are one of the developing countries in the world. So in last, I conclude, conclusion is science and technology has profound impact on all of humanity activities. Science and technology inventions and discoveries include theory of evaluation and discovery of genes and have given humanity related to human existence. Science and technology have had an immeasurable influence on the formation of an understanding of the world, our view of society, and our outlook on nature. Thank you so much.
Okay, thank you so much. Now I would like to request those participants who was suffering with poor network connection, like Chama Mishra and Anamika Rajvanshi. Are you here, Chama?
Yes, sir.
And Anamika? Okay. Aditya, are you here? Aditya Kumar Singh. David Samuel. Pallavi, Anisha Tiwari, Siddharth Singh, Ayushi Panchal, No. Okay. So now, Chama, you should share your screen.
Okay, sir.
I have made you the co-host. You can share now.
Yes, sir. My screen is clear.
Yes, yes. Your screen is visible.
Okay, sir. My name is Chama Misra from Bansal Institute, second year. All teachers and my dear friends, all of you, good afternoon. We represent our presentation on the National Science Day. Please remember the discovery of the Raman effect. We also we remain on noted the novel, and we also our presentation. We use of all wasted material as like dustbin, dust and dust particle and food material who not use our daily life, then or then use we participate other organic material used in agriculture, and the other waste materials, cow dung and urine are collected and in uh, recycling biogas used for LPG and CNG, and other waste material as like factories carbon dioxide monoxide who is released the factories are recycling use the thermal energy, and other material are which is not used in our life in recycling and waste material in our daily life. So in you so these using in useful in our life. Thank you, sir.
Okay, thank you. Thank you so much. Now, Sandeep Srivastav. Sandeep. Sandeep Srivastav.
Yes, I am audible.
Yes, yes, you are audible. I have made you co-host. Kindly send me some details like you are UG or PG student?
Sir, I am research scholar in Galgotias University.
Okay,
Greater Noida.
PhD student. Okay.
Yes, yes.
Okay. Now, please send your email ID to me, and you can. What is your topic of presentation?
Sir, I am just going to present my topic related to wireless sensor network solution for precision agriculture.
Okay, so you can share your screen, please.
Okay, sir. After that, sir, I got your number. I share my email ID.
Okay.
And all details.
And that is required for the announcement, for the certificate and all the things. So, okay.
Sure, sir. Just a second.
Now,
I'm going to
yes.
share. Just my screen is visible.
Yes, your screen is visible.
Okay, sir. Okay. Just make it full screen.
In the below, you can find an icon.
That's good, sir.
Yes.
Okay. Thank you, Lord sir, for the last moment you will give me the permission to allow me to show my presentation. First of all, So
Our pleasure. Kindly
thank you,
continue.
sir.
Yes.
Okay. So today is the National Science Day that is celebrated annually on February 28 to commemorate the discovery of the Raman effect. Raman effect, that means Dr. Sir Chandrasekhara Venkata Raman, an Indian scientist, announced the discovery of the Raman effect on 28 Feb 1928. So Indian scientists were awarded the 1930 Nobel Prize in the physics for his work on the scattering of light and for the discovery of the effect named after him. The Nobel Prize organization said on its National Science Day. So basically, I am coming here with my research domain area, wireless sensor network. Nowadays, it's a big deal with the agriculture because in our country is based on the agriculture requirements. So basically, wireless sensor network have attract much attention in recent years in agriculture work. So potentially, application of the wireless sensor networks are used for collecting, storing, and sharing sensor data. These data work for various applications like monitoring the agriculture and its weather forecasting that is used in precision agriculture application. So where real-time data of climatologically and other environmental properties are censored and controlled, decisions are taken based on it to modify them. So architecture of a wireless sensor network system comprise for a set of sensor nodes and a base station that communicate with each other and gather local information to make global decision about the physical environment. So in this scenario, precision agriculture can be defined that is the art of science of using advanced technology to enhance crop production and monitoring the weather forecasting. So WSN is a major technology that drives the development of precision agriculture. The science engineering always questionnaires, making the questionnaires for association of this agriculture type center around increasing the efficiency to in such a sustainable manner. So increases in agriculture efficiency will use for the networking sensor to important the integration of their rootness, their stream, so that display the record information and actual for the human take the precaution before getting the any hazardness. So concept of the precision agriculture has been around for some time now, and there are so many researchers worked for that. So if I'm taking one example here in California, they are designed, the scientists design, deployed, and analyze output of a large scale implementation of a wireless sensor network that applied the technology to discover the protocol features for the agriculture. So in this scenario, there are so many things that are required for making this safety purpose and effectiveness agriculture monitoring concern for both system level issue like unattended operation, maximum network lifetime, adaptability of even functionality. So these are making the giving the fruitful result for the precision agriculture. Now, if we are talking the conclusion, so these precision agriculture and wireless sensor network application combine and provide a new era of the research that will greatly improve quality in agriculture production. So irrigation will have dramatic reduction in the cost needed. And furthermore, we are finding the optimal sensor topology that take the lower implementation cost. So the general and common person or farmer can absorb this technology very easily and apply the solution for all kinds of field and cultivations. That's all. Thank you for listening me.
Thank you. Thank you so much. So I am again repeating the name of absent student, Aditya Kumar Singh, David Samuel, Miss Pallavi, Anisha Tiwari, Siddharth Singh, Ayushi Panchal. Okay, so no one present here. So we are proceeding for the next session, that is quiz. Dear participant, I will share a link in the chat box, and after 5 minutes, 5.5 second, 5 minutes 5 second, I will close the feedback, quiz form link. It will not accept. So please be careful and submit your quiz in the stipulated time. I am going to share the link at 1:30, and you have sufficient time. I will close the link at 1:36. So you have sufficient time to do your quiz. After that, we will calculate your marks and will announce your name. So we remain with us. There is a feedback link also, which will be also shared, that is utilized for the generation of your certificate. So do not leave this meeting without attempting the quiz and feedback form. Okay, so this is I have shared the quiz link. Just click on the link and start the quiz. Everyone get the link of the quiz?
Excuse me, sir.
Yes.
Sir, I'm not able to open the link. When I'm opening the link, it shows other language. I'm not able to understand what is this language.
In another language? How it is possible?
I'm not able to write it in English. At least it's something South Indian language it's showing over here. I'm not able to understand what it's saying. I can show you the screen also, what it's saying.
Okay. Anyone other facing any difficulty? Actually, check your setting, language setting, because I have tested this quiz on various laptops. Ashish sir?
Hello.
Yes, Rajesh sir?
Yes, sir.
Are you able to take the quiz?
Yes, sir, I can do.
Okay, thank you, sir.
Sir, where we have to give quiz?
I have given the quiz link in the chat box, sir.
Okay, okay.
And it will be closed at 1:36.
I'm sorry, sir. I'm not able to change the setting. It's showing by your side and it's in the South Indian language. I'm not able to translate into the English. So maybe
How it is possible?
I don't know. You can make me co-host. I can show you the by the screen presenting.
Okay. Okay.
I'm presenting screen. If you make okay, I'm co-host. I'm presenting screen. You can show my screen.
And this is due to what?
I checked my computer language. It says in English, but after that, I'm still
No,
not.
no, you are using Safari.
I'm not using. Yes.
You should. Do you have Chrome or any other browser?
Yep, I have Chrome.
Then you can open this link in the Chrome.
But it's not
just open the Google.
Okay, thank you so much.
And this error due to from your side.
Okay.
Because we have already tested on various computers. I am giving you 1 minute more. Now the time is over. I'm going to close the, please submit. I'm going to close.
Sir, excuse me, just give us few more minutes because it was difficult to open at the right time. So
3 minutes more exactly to be
No,
precise.
no 3 minutes.
Okay, thank you, sir.
Please wait, sir. Please, sir.
Please try to submit as earliest. I have given 2 more minutes. Got 25 response only. Please submit. We have given 10 minutes. Should I close?
Yes, sir.
Okay.
Yes, sir.
Ma'am, who is facing? Disha ma'am?
Yes, sir.
Yes. Okay. Have you submitted?
Wait, sir, please.
Okay. The quiz consists very basic question for the science. So now I'm going to stop the quiz. Please submit. Okay. Thank you. Thank you so much. Now the result is also
Sir, I am not able to submit it. Just a few
actually
seconds.
I have announced. Okay.
No,
So
no, few seconds. Just a few seconds is moment, sir.
Okay. Please submit.
Sir, please, can you please give me some time?
Ma'am, I've already given.
सर एक्चुअली नेटवर्क बहुत स्लो चल रहा है, सो ज्वाइन नहीं हो पा रहा था, इसीलिए थोड़ा सा टाइम लग गया. अभी जस्ट ज्वाइन हुआ है मेरा.
but ma'am.
Only 5 minutes.
Okay.
Okay, thank you, sir.
Okay. Okay, now I am going to close because the quiz was open for the 5 minutes only as per the rule, but I have given 15 minutes for the quiz. Now please submit.
Sir, we are already submitted.
Okay, thank you, thank you. Okay, and now I am going to
Thank you,
close.
sir.
Please submit.
Yes, submitted, sir.
Okay, thank you. Now quiz is now closed. Now we can't open the quiz again. Okay, thank you so much for the participating in the quiz. Now I'm announcing the winner, and there was 25 question, but no one achieved all the question, all marks, all the answer. The highest marks is 22 for the Riddhi BTech CSC Galgotias University. The second one Subra Jyoti. Third one is Devans Misra from BTech CS. Okay, for the quiz competition. First Riddhi, second Subra, third Devans Misra. For the poster presentation, again in the UG we have categorized 3 type of entry. For the Subra Jyoti, it is 28, again winner in the UG student category. The Disha Gupta, UG student, 26 marks they have got in UG category. That and comes at the second position, Disha Gupta. The third position, Chama Misra, UG student.
Thank you, sir.
In the faculty category, the only one faculty was there. So Dr. Rajesh Yadav is the winner and secured 29 marks. The Shweta Misra is the PG student, was the winner in the PG student category. And in the PhD scholar category, Sandeep Srivastav is the winner. Now again, I am announcing in the UG student category.
Thanks, sir.
The Subra Jyoti won. Second position Disha Gupta. Third position Chama Misra. Dr. Rajesh Yadav in the faculty category. In the PG student category, the Shweta Misra. And in the PhD scholar category, Sandeep Srivastava.
Thank you, sir.
Thank you, sir. Thanks a lot.
Okay, thank you so much. And now I am going to share the feedback link. Kindly provide your feedback for the session, and thank you so much for making this event successful, for joining us. Okay, please find the feedback link. Thank you, Ashish. Are you here? Actually, Ashish have sent the quickly sent the result. That's why we can compile the result. And another faculty also submitted the result. So on the basis of 3 evaluators, we have reached on this decision. If anyone has objection, we can also solve. They are welcome. So kindly provide your feedback. Thank you so much for being with us. Thank you so much. So everyone got the feedback link. Please provide your feedback that will be utilized to generate your participation certificate.
Yes, sir.
Okay, sir. Yeah, got it.
Okay. And we will send the certificate to winners. Apart from the participation certificate also, Thank you so much.
Thank you, sir. Thank you for